is Zoe Smith, and my protein was on Piezo 1 argine 2456 and replaced by histidine. And this is my project. My model is made of Play-Doh, marshmallows, and paper for like the little legs, and it's shaped as a fidget spinner to show all like the properties and stuff. Piezo 1 plays a really big role in translating mechanical stimuli to biochemical signals. It's primarily involved in sensational of mechanical stretch, pressure, in response to force. Piezo 1 primarily functions as a mechanosensitive receptor that converts its mechanical forces into cellular electrical signals by allowing ion flux. Its role in medicotransduction and regulation of the ion flow justifies its categorization as a receptor, with the key distinction as its ligand is mechanical rather than chemical. This protein is usually found in subcellular locations such as plasma membranes. The cells that it's usually found in are endothelial cells, red blood cells, sensory neurons, smooth muscle cells, keratinocytes, chondrocytes, and many more. It's found in a wide range of animals, including mammals, fish, amphibians, invertebrates, and is conserved across these species, reflecting an essential role in mechanotransduction. This mutation causes the protein to be less active because it might not respond effectively to mechanical forces due to structural change induced by the substitution of histidine for argine. This mutation might also cause some structural changes affecting the stability of the protein's overall structure. Argine is a larger side chain and might contribute to more stability in certain regions of the protein compared to histidine. It might have a less rigid structure, potentially affecting the conformation of the proteins in its activation state. But it really can be more active or less active depending on where the change has taken place, responsible for the mechanical forces. The histidine mutation could alter the electrostatic interactions affecting the gating mechanisms of the channel. In general, mechanosensitive channels like piezo tend to become less active with mutations that disrupt their gating and structural integrity. But there are less cases where such mutations can make the channel more sensitive to mechanical forces. This is a hydrophobic polar protein because of its ability to form hydrogen bonds and ionic bonds with interactions. It is also positively charged with the physiological pH. The functional groups of the argine side chains, like the AA chains, are responsible for basic nature of argine, and it's responsible for its ability to form a strong electrostatic interaction within the protein with surroundings of molecules. The positive charge on the G group in particular is more likely and important to piezo 1's mechanicosensitive function, influencing its conformational changes and responses to mechanical stress. The mutation of argine 2456 to histidine and piezo 1 alters the side chain's ability to participate in electrostatic interactions, potentially making them less stable and more sensitive to environmental changes. This could impact the protein's gating dynamics and overall functioning activity, depending on the role that of the residue in piezo structure. This is a neutral protein. It's constantly at a state of equilibrium, so it's not positively or negatively charged. compared to the other 19 AAs, it is considered to be a medium size. Like, it's not too big and it's not too small. And lastly, just the overall impact would depend on the exact role a residue's piezo structure would function. So, yeah. In conclusion, piezo bonds protein plays a critical role in the mechanical transduction, enabling cells to respond to mechanical stimuli, such as stretch pressure. The mutation introduces notable changes in the protein's functionability, primarily throughout alterations in the electrostatic interactions, hydrogen bonding, and structural stability. Argon-stable positively charged and guanidines groups facilitate strong interactions, and they are key to piezo 1's gating and mechanical sensitivity, while histidine's pH-sensitive amylase ring introduces more dynamic and potentially less stable interactions. This could result in altering the gating behavior, making the protein either more or less responsive in mechanical forces depending on the environmental context. The mutation underscores the delicate balance between structural and function, and the consequences of such change can have a significant effects on cellular responses to mechanical stimuli and broader orgasmal function, particularly in the tissue where medico sensitivity is critical. Understanding these changes can provide valuable insight into the role in which and its potential implications for health and diseases.